Imagine you are treating a patient with advanced stage glioblastoma. The brain tumor is surgically removed and the patient is given a regimen of chemotherapy, but still he has rapid progression of his cancer. The tumor has become too widespread for more surgery or radiation to be helpful, and it is so heterogeneous that no therapy exists to target it. We need a new approach, and systems biology may get us there. But why systems biology? And more specifically, why should we use systems approaches in cancer research? To answer these questions, we first need to talk about three things. First, we'll define systems biology. Second, we will talk about how cancer is considered a complex system. And third, we will investigate how systems approaches are used in cancer research. Together, these tell a story about why systems approaches are useful to cancer research and have the potential to be useful in the clinic. So what is systems biology? Systems biology is a field that has been notoriously hard to define. The word was first coined in the context of a conference in 1968, but it took a while to gain a foothold in biology due to doubts that computational methods could be used to understand living systems. In the 90s, three things happened. The Human Genome Project began, and high-throughput methods began generating unprecedented amounts of data. And by then, computers were becoming more powerful and ubiquitous. Systems biology became gradually accepted as a new discipline. Then in the 2000s, the Institute for Systems Biology and several departments and universities across the country were formed, helping to solidify the discipline even more. But still, systems biology was lacking a cohesive definition. Despite this, over the last few decades, the field has exploded, with more and more papers being published every year. So how do we define systems biology today?
To me, systems biology in general is asking a question or defining a problem that at the onset, by definition, is beyond one's intuition.
Systems biology means studying a problem, a biological problem that is so complex that you can't just intuit the answer. You really need the benefit of computation, math, in order to dissect the parts and understand how they work together.
Systems thinking generates non-intuitive solutions because complex systems are more than the sum of their parts. In cancer in particular, many interacting parts can lead to unexpected outcomes, and we need systems biology to predict such outcomes.
Systems biology, to my mind, involves sort of modeling the complexity in general, but when we're talking about cancer, sort of modeling in particular the signaling complexity within a given cancer.
So systems biology is definitely, for me, the idea of trying to understand a biological system, whether it's an individual cell, whether it's how cells come together, whether it's how cells come together into organs, how organs come together into people, or how people come together into larger networks. And trying to essentially measure information about all of those pieces and then use approaches to learn about what all of those pieces mean emergently, or something that you couldn't have understood by measuring one of those things in the absence of the other things at the same time.
Systems biology is the marriage of computational models with sophisticated experimental data as part of that iterative process of predicting and testing.
Altogether, we might say that systems thinking is considering that the whole is more than the sum of its parts, and systems biology is using this thinking to evaluate non-intuitive behaviors of complex biological systems at multiple scales with computational modeling and experiments. In all of these definitions, one idea has come up a lot that explains why cancer is so hard to treat: complexity. So you might be wondering at this point, what makes something a complex system? Systems thinking is particularly useful when dealing with complexity. Plenty of problems are complicated or hard to solve, but complexity is something different, and understanding it can help us figure out why cancer is so hard to treat. Let's turn to an example where modeling a complex system has already made a huge difference to humankind: hurricanes. Weather modeling has been somewhat of a success story for systems thinking, and can give us an idea as to what success might look like for systems biology and cancer. To understand the difference between complicated problems and complexity, imagine a fan blowing in an office. This is a simple scenario, and it would be easy to understand how the fan will affect other things in the room. For example, a pencil on the desk might roll off due to the wind, and it will fall on the floor. Importantly, we can predict what happens based solely on the underlying principles of physics. We can intuitively understand where the pencil would end up without needing to go buy a fan and check for ourselves. This is a complicated rather than complex problem. How do we go from this simple system to understanding and predicting the movement of a hurricane? Imagine now that I could give you information on the prevailing weather conditions like temperature, humidity, or large-scale wind patterns. With this information, could you predict where a hurricane would form? While you might have some ideas based on patterns of past hurricane formation, it would be difficult to use intuition alone to find an answer. By the way, there's a hurricane forming here. The answer is surprisingly difficult because hurricanes arise in the complex system of weather. Complex systems contain several simple components, like air molecules, that interact in nonlinear ways to produce unexpected behaviors or emergent behaviors. These are patterns that the whole exhibits but individual parts lack, and hurricanes are one such example. Importantly, it is not possible to calculate where a hurricane emerges or ends up solely using equations from physics, the way we could for a pencil rolling off a desk. To predict the formation of a hurricane, the event must be simulated. We must make a model that matches reality as closely as possible, and then watch as the model evolves. Through systems modeling of the weather, we are better able to predict what the course of a hurricane will be. For example, in 2005, Hurricane Katrina devastated the southern United States, particularly New Orleans, in part due to high uncertainty in the predicted track of the hurricane. Only 10 years later, weather modeling improved enough to reduce the margin of error in the prediction by 40%. And today, forecasting models are even better. In this case, better modeling leads to better predictions and can save lives. Just like hurricane modeling, modeling cancer as a complex system can help save lives. Cancer is a complex system because a relatively simple event, a mutation, leads to individual components, the mutated cells, that can interact in non-intuitive ways. These interactions give rise to unexpected emergent behaviors such as metastasis, heterogeneous phenotypes, and therapy resistance. Just like weather systems, where complexity exists at a local and global scale, it's important to think about complexity in cancer on multiple levels, from genes to patients. Let's consider how some of these components interact. At the molecular level, components might be proteins in a signaling cascade.
These cells are driven by numerous pathways that are typically cascades where lots of proteins are talking to each other.
There's so many things out there that cells could respond to. There's so much internal machinery, the way that they could transduce that information.
We attempt to try to understand what all of those molecules are doing and to try to understand how all of those pieces actually connect together in order to make a decision.
And so understanding those processes, how those cancer cells take those substrates, those resources from the environment is very complex, and we need computer models to try to understand those processes.
At the cellular level, different cell types interact and can cause drastic phenotype changes.
We're interested in the biochemistry of how cancer cells make decisions. For example, how a cancer cell decides should it die or not in response to chemotherapy or targeted therapy.
There's also in a cancer like glioblastoma incredible heterogeneity, and so it's not sort of monolithic where it's like, oh, one mutation drives it. There's just too much happening. I've heard it described as like whack-a-mole, for example. Sort of, you smack one target and, two other drivers of the cancer can pop up to compensate.
Hopefully you are already getting a sense of why systems biologists believe modeling cancer as a complex system is needed to make sense of this disease. Therapies today often don't help everyone, or they lead to relapse because they treat cancer like a complicated problem rather than a complex one. Modeling complexity at every scale can help us predict when certain patterns occur and how to prevent them, leading to lives saved. So cancer is a complex system, and those could usually benefit from systems approaches. How is systems thinking applied in practice? Let's consider a specific problem with cancer, the fact that it is constantly evolving.
One of the reasons cancer is so incredibly hard to treat is that there's essentially evolution happening in the cancer. And whatever we throw at a given cancer, the cancer can very quickly evolve around whatever we're throwing at it.
This evolution to a treatment-resistant disease may counterintuitively be promoted by aggressive therapies. Reducing the intensity of the therapy could also reduce the pressure for cancer cells to adapt.
there might be smart ways revealed by computation that you can schedule drugs to achieve the desired effect clinically for a patient, but to minimize toxicity.
There's the potential for systems biology to change the conversation away from the decades-long view of eradicating the disease, cutting out the cancer and killing everything else that wasn't removed. I think there's a potential to incorporate ideas of feedback, adaptation, competition in the way that we deploy therapies to patients.
One systems biologist who has been working to change the conversation is Robert Gatenby, who has developed an approach to cancer treatment called adaptive therapy.
Adaptive therapy is meant to increase the overall survival using existing drugs and lower doses to improve the quality of life.
To maximize the time it takes for cancer to evolve resistance to treatment, Gatenby says you need to consider evolutionary principles.
And this way for 50 years. Standard therapy is maximum tolerated dose continuously until progression. The problem is evolutionarily, that's usually unwise. And the reason is that by applying the same treatment over and over again at maximum tolerated dose, what you're doing is maximally applying evolutionary pressures for resistance, and you're also removing their competitors.
The key insight with adaptive therapy is that the proliferative drug-sensitive populations can prevent smaller resistant populations from growing. Killing the sensitive cells in normal chemotherapy therefore selects for resistant cells, and they can proliferate because their competitors, the sensitive cells, have been killed. This is called competitive release, which is avoided in adaptive therapy.
A typical adaptive therapy approach would be to say, instead of giving maximum tolerated dose just continuously, to give a reasonable dose of therapy, just enough to knock the tumor down. And in this case, you explicitly leave behind a significant population of sensitive cells. The sensitive cells are your buddies. You can control them, and you want to use them to control the tumor cells you can't control. You can then stop therapy, and the tumor will regrow. But you'll notice that as it's regrowing, you're not putting any selection pressure for resistance.
So the sensitive cells regrow to form the same mix of resistant and sensitive cells as before treatment. The tumor is then controlled by another round of treatment.
The amount of therapy depends on what's happened to the tumor. If the tumor has grown a little bit, you give higher doses. If it's gotten smaller, you give lower doses. And you simply just keep going through this iteration. What was particularly interesting in the preclinical studies is that what we found was over time, we could maintain control of the tumor using lower and lower doses of drug to the point where we could almost not treat at all.
This approach is making its way into the clinic today, with the first clinical trial focused on treating castrate-resistant prostate cancer with abiraterone.
And that's been published, and the results were really quite good. Overall survival was increased by almost 2.5 years. Now we're going to be using androgen deprivation therapy with abiraterone, and we're going to be using this with mathematical models where the dosing is changed at every treatment based on the model design. So that would really be the most sophisticated mathematically and evolutionarily trial that we've tried so far.
If you look closely, you might notice a loop between computation and real life that we touched on before. This is a hallmark of the systems approach. Data from the patient is used as input to a computational model, which can make predictions about cancer progression. In the lab, these computational predictions are then tested with experiments, generating knowledge, validating the model, and providing more data. When a systems approach translates to patient care, these same models can be used to predict the behavior of a cancer and influence treatment decisions. The patient can then be monitored, generating new data for computation. The iteration between computation and patient data can help tackle problems like therapy resistance that arise in cancer treatments today.
When we move away from thinking of a one-size-fits-all kind of approach to treating disease, I think that can be powerful.
It provides for me hope that there are ways to unlock extreme sensitivity to targeted therapy in combination with chemotherapy And I think systems biology is probably the way to get there.
Now there's one last question we would like to answer. How could a clinician get involved in systems biology?
Even amongst the clinician scientists like me, there are very few of us who are doing systems biology work ourselves. So really, I think the best way is to connect and collaborate with systems biologists on the research side and really work to apply those techniques to what we're doing in the clinic.
No one can know everything. And the oncologists can work with mathematicians or with bioinformaticians and that sort of thing. And it's a beautiful thing if it works, because you learn from each other, you respect each other, and you contribute to each in its own way.
There's a lot of individuals that the first thing that they say when they come and talk with me is that I'm not a systems biologist. And my reply these days now is to say not a systems biologist yet. Anybody who is willing to learn new things and ask questions that maybe they fear are stupid, but almost certainly are not, has the potential to grow or begin incorporating systems biology elements, systems biology thinking into their own work.
Remember that patient with advanced glioblastoma who had no treatment options? Now let's imagine a world in which oncologists use systems approaches to diagnose and treat patients. After surgical resection, you sequence the tumor and determine which therapy regimen would best fit the patient's genetically complex cancer. During the course of treatment, you measure biomarkers and use computational models to adjust the treatment plan as necessary to avoid relapse. The continuous feedback between modeling and patient data gives your patient the best chance of survival, and the glioblastoma responds to this evolving regimen. The patient's life expectancy climbs dramatically, and the use of adaptive therapy means the patient doesn't relapse. Systems biology plays an essential role in making this dream a reality.